Right, some more terminology that we'll need for this chapter. First one is phenotype. And phenotype is what something looks like. That's the characteristic that you observe or that you see. An example of that would be these giraffes. We've got an albino giraffe. That would be its phenotype because I'm describing what it looks like. And normal giraffes. A related but slightly different concept is the genotype. And that's the underlying genetic composition. So what genes are they actually carrying? So both of these giraffes look normal, but they might be hiding a recessive trait like albinism, even though they look or have a normal phenotype. Another thing when you're doing heredity, a lot of times you look at it because you want to analyze and predict outcomes. You want to know the possibility of these two giraffes having an albino offspring, or of two people having an offspring with a particular characteristic. To make some of those predictions, you can use a Punnett square. And in a Punnett square, you represent your genes or your characteristics with letters. So instead of writing white or albino and normal every time, you just use capital and lowercase letters. The capital letter represents the dominant gene, so by definition, if it's a capital letter, it's a dominant gene. And in this case, with albinos and normal giraffes, the normal color is dominant, so it's got the capital letter A. Albino is the recessive trait, so it can hide or get covered up, and so that's got the lowercase letter A. If we walk through a P, F1, F2, kind of a classic Mendelian monohybrid cross, we have normal and albino, and those can be represented by true breeding, two capital A's, and true breeding, two small a's. This would be homozygous dominant, so we've looked at dominant and recessive. We've also looked at homozygous and heterozygous. Having two capital letters makes you homozygous dominant. Having two lowercase letters makes you homozygous recessive. If we cross these and we use a Punnett square is just a grid where you put the parents on the outside. So I'll put the normal parent on the side out here and the albino parent across the top, and then you'll pull their letters into the center of the square to represent their offspring. So this big A will go across, and this big A will go across. This little a will come down to fill in the columns, and that little a will come down to fill in. And that's a Punnett square. So I can see in my P generation, if I have a true breeding normal and a true breeding albino, homozygous dominant and homozygous recessive, if I cross them, all of the offspring are going to be heterozygous. So my F1 are all heterozygous, and they look, their phenotype, what I can see, they look normal. So they have a genotype of big A, little a, but the phenotype is normal. If I cross two of my F1s together to give me F2, and I'll just write it over here. So if I cross one of these with a second F1, and put that into a Punnett square. Again, one parent down the side, so capital A, lower a. One parent across the top, capital A, lower a. This A will come down, the other A across and down, down and across, and down and across. So what I end up with is one, in this case baby giraffe, that's homozygous dominant, with the big A. I get two of them, this one and this one, that are both heterozygous. So I have two that are big A, little a, heterozygous. And then I get one that's homozygous recessive, two little a's. And that's a classic pattern that you see. If you're looking at the genotype, or what's in the DNA, it's 1 to 2 to 1. But remember, anything with that big A, because it's dominant, looks the same. So what I see in the phenotype is a 3-to-1 ratio. 3 normal to 1 albino. And this is a figure from your book. It shows this. So our homozygous albino mother, 2 little a's, homozygous pigmented or normal father, and our F1 are in the square. So all of them are going to be heterozygous with big A and little a. All of them are pigmented or normal colored. If we cross 2 of these heterozygotes, so a heterozygous mother, a heterozygous father, we get 1 homozygous dominant and 2 heterozygous offspring, but they all look normal because they're all pigmented. And then we get 1 that is albino or homozygous recessive. So 1 to 2 to 1 or 3/4 to 1/4, depending on if you're looking at the genotype or the phenotype. When you're using genetics, it's all about probability and statistics because it's random exactly where the chromosomes go and exactly which egg is fertilized by which sperm. So it's all about probability. Very similar to rolling dice or tossing coins or dealing cards. Those are all probability based. Same thing is true with genetics because it's all random events. But you can make predictions with that. So staying with our giraffes for a minute, if we have an albino giraffe, and this one happens to be a female giraffe, when she makes eggs, that albino gene, because that's all she has to give, both of her eggs, or all of her eggs, 100% of them, are going to have that albino gene. So the 1 represents 100%. It's just moving the decimal. So you just move the decimal 2 places for the percent. So 1.0 means 100% of her eggs carry albinism. If you cross her with a male giraffe that has a normal and an albino gene, so he's heterozygous, he's going to make a normal sperm, an albino sperm. Because we're interested in albino giraffes, half of the sperm are carrying that little a, so 50% or 0.5. To get the chance that an offspring is going to be albino, you take the possibility in the mom multiplied by the possibility of the dad passing it on in its sperm. That gives you 0.5 or 50% chance that these 2 giraffes would give you an albino offspring. That might be important if you're running a zoo, but we're probably more interested in a human characteristic. So let's look at a human example. If parents are heterozygous, so they're each normal but hiding the gene for Tay-Sachs, what's their probability of having a child with Tay-Sachs? If you have Tay-Sachs in your family and you might be carrying it, this is a very important question. Tay-Sachs is an awful, awful illness. And in Tay-Sachs, babies are born perfectly normal. They look normal, they act normal, they develop normally. And then sometime within the first year or so, usually about the time after they start saying their first word, maybe they've taken a couple of steps, things like that, you start to notice a regression in their behavior. So they'll start to lose the ability to say those few words that they know. They stop being able to walk or kind of control their movements quite as well. Because with Tay-Sachs, what happens is kind of proteins build up and it has an effect on the brain. It's a neurological disorder and it's degenerative. So the babies gradually, lose their words, lose the ability to talk, and revert back. Eventually, they normally die by the age of 5. And it's a gradual, slow, sometimes painful disease in the offspring, and also for the parents to watch your baby be normal and then gradually die, and there's nothing you can do because it's in the DNA. So that's a really important question, and you want to be able to make predictions on that. So if the mother has a normal gene and a Tay-Sachs gene, she'll make eggs. Half of the eggs will have the normal and half will have the Tay-Sachs. So she's got a 0.5 or 50% chance of passing on that Tay-Sachs gene. If her partner also is heterozygous, so he'll make half normal sperm and half Tay-Sachs sperm, again a 50% chance of passing that on. You multiply the 0.5 times 0.5, so that baby has a 0.25, or any of their offspring has a 0.25 or 25% chance that this couple will have a baby with Tay-Sachs. When you're working with things like giraffes, or in this case crocodiles, or plants, you can do a test cross to figure out if the individual you're looking at is homozygous dominant or heterozygous, because they look the same, right? So you can do a test cross. In a test cross, one parent is always going to be homozygous recessive, and the other parent is unknown. If we're test crossing for our white alligators here, we have a homozygous recessive white alligator. So 2 little letters, M, and we have a father, and we don't know. Does he have 2 capital M's or a capital M and hiding that lowercase m? So we do a test cross. So we cross them, and if the unknown, so if the father over here, if he has 2 capital M's, when we cross it, all of the offspring will get a capital M from dad, and all of the offspring will get a lowercase m from mom. So all of them will be heterozygous, and they'll all be pigmented. So none of them will be white alligators. However, if that unknown, so if dad over here has capital and lowercase, so it's hiding the lowercase, then half of them will get a capital M from dad, and half of them will get a lowercase m from dad. Mom will only give lowercase m to all of her offspring. So in that case, you will get 2/4 will be heterozygous, and they'll be pigmented, and 2/4 will be recessive. So you'll get half and half. Half of them will be normal, and half of them will be white. And you could do this with flowers or anything else. The main thing is that one parent is this homozygous recessive, and the other is unknown. Albinism is kind of an interesting trait, and it's found in pretty much all animal species. So we've looked at the albino giraffes already in this chapter. There are albino deer, albino crocodiles. There are even albino humans. And in normal populations, the rate of albinism is about 1 in 20,000 people will be born as albino. However, there is one group, and it's pictured here. It's a group of Hopi Indians that live in the Black Mesa area. It's kind of out in Arizona, where their frequency, instead of being 1 in 20,000, it's 1 in 200. So that's 100 times as frequent as most other populations. And the reasoning behind that, most of the time, if you're albino, historically, they would have been kind of outcast. They look different, so kind of pushed to the fringes of society. However, in the albino culture, if you were, or in the Hopi culture, if you were born albino, they were considered to be very pure and clean individuals. They often assumed positions of leadership. They became chiefs and healers and religious leaders. So they had a social prestige, and so they didn't have to worry about being ostracized and kind of pushed to the margins of their community. Also, these Hopi tribes had small farms. So during the farming season, where they had to go and tend to their fields, usually it was the men who left the camp every day and went out to tend the fields. And the women would stay back in the camp and do other chores. Well, albino individuals are very light because they don't have any pigment in their skin or eyes. So if albinos go out in the sun, they sunburn very quickly. They also have trouble seeing in bright light, and they get cancer very easily because of the no protection in their skin from the sun. And this tribe of Hopis allowed men who were born albino to stay in the village and do other tasks instead of going out to the fields. This helped them deal with any problems they would have had being albino. It also gave them a substantial mating advantage because they were the only ones in camp all day during the farming season, so they were likely to father offspring. Also, it was a small group, and so over time, small groups tend to increase in interesting or rare alleles. So, one other case of a possible albino, and you guys may recognize this. This is a kind of cartoon depiction of Noah's Ark, and Noah may have been an albino. And I'll read something here, look at something that discusses was Noah an albino? So the biblical character of Noah, along with the ark and its animals, is among the most recognizable figures in the book of Genesis. His birth is recorded in a single sentence, and although the story of how the ark was built and survived the great flood is told later, there's no mention of Noah's physical appearance, but other sources contain references to Noah that are consistent with the idea that Noah was one of the first albinos mentioned in recorded history. The birth of Noah is recorded in several sources, including the book of Enoch the prophet, written about 200 BC. This book, quoted several times in the New Testament, was regarded as lost until 1773, when an Ethiopian version of the text was discovered. In describing the birth of Noah, the text relates that his flesh was white as snow and red as a rose, and the hair of whose head was white like wool, and long, and whose eyes were beautiful. A reconstructed fragment of one of the Dead Sea Scrolls describes Noah as an abnormal child born to normal parents. This fragment of the scroll also provides some insight into the pedigree of Noah's family, as does the Book of Jubilees. According to these sources, Noah's father and his mother were first cousins. His father was the son of Methuselah, and his mother was the daughter of Methuselah's sister. This is important because marriage between close relatives is sometimes involved in pedigrees of autosomal recessive traits like albinism. If this interpretation of ancient texts is correct, Noah's albinism is the result of a consanguineous marriage, and not only is he one of the earliest albinos on record, but his grandfather Methuselah and Methuselah's sister are the first recorded heterozygous carriers of a recessive genetic trait.